All right, today we're diving into a really frustrating mystery. Why do these life-saving cancer treatments sometimes just stop working? It's a huge puzzle, right? And the clues, well, they're hidden deep inside our own cells. So let's play detective.
So take a look at this number. That's nearly 700,000. That is how many lives were lost to breast cancer just worldwide in 2020 alone. I mean, that's a staggering figure. It really shows you the scale of the challenge we're up against.
And for a long time, chemotherapy has been one of our absolute best weapons in this fight. We're talking about drugs that are designed to be ruthless, right? They seek out and destroy cancer cells. And honestly, they've saved countless lives.
But there's a really frustrating catch. Sometimes the cancer cells, they actually learn, they evolve, and figure out how to survive the treatment. This is what scientists call chemoresistance, and it is the huge mystery we need to crack. So, what is really going on inside these cells?
Okay, so our investigation starts with our first big clue, a glitch. A major glitch in how cells divide. This one takes us right into the heart of how cancer cells multiply, and you'll see how they turned their own internal chaos into a pretty powerful survival advantage.
Now, to get this glitch, we got to look at something called the centrosome. The slide calls it the air traffic controller, and that's a perfect way to think about it. It's basically the director of operations for cell division. In a healthy cell, its job is to make sure everything is smooth, orderly, and happens exactly right every single time.
See, here's how it's supposed to work. A normal cell has exactly 2 centrosomes. That's the magic number. It lets it divide perfectly into 2 healthy cells. But aggressive cancer cells, they often have way too many. Now, this should be a red flag, right? It should trigger the cell to self-destruct. But they've got this incredible, clever trick. It's called centrosome clustering. They just bundle all the extras together to pretend everything's normal, and they cheat death.
And look, this is not just some neat little trick. It's an absolutely key survival mechanism. By clustering those extra centrosomes, the cancer cell dodges what should be a fatal error, and it just keeps on multiplying. This kind of chaotic division, it's a hallmark of the most aggressive cancers. And that is our first major piece of the puzzle.
All right, on to clue number 2. And this one is all about the cell's tiny little motors. Yeah, you heard that right. We're talking about a totally different cast of characters here, a team of microscopic engines that do all the heavy lifting inside the cell.
These guys are called microtubule motor proteins, or MIMOs for short. And the best way to picture this is to think of a cell as a huge, bustling city. The highways are these tracks called microtubules, and the MIMOs? Well, they're the tiny trucks driving on those highways, constantly moving vital cargo around to keep the whole city running.
So these little motors, they basically come in 2 main families that just move in opposite directions. You've got kinesins, which head toward the plus end of the highway, and you've got dyneins, which head toward the minus end. In a normal, healthy cell, this whole thing is a perfectly coordinated ballet, a beautiful logistics operation.
And this is the crucial discovery. Researchers found that in these chemo-resistant breast cancers, the levels of these MIMO proteins are all out of whack. It's like the cell's entire shipping and receiving department has gone completely haywire.
Okay, so let's connect the clues. On one hand, you have this glitchy, chaotic cell division. On the other, you have these faulty little motors. And this is where researchers started to wonder, wait a minute, are these really 2 separate problems, or are they 2 parts of the same story?
I mean, was this all just a coincidence? Or could the faulty transportation network actually be the cause of all that chaos during cell division?
This question led to a really groundbreaking hypothesis, which was laid out in a recent scientific review. Let's put it in simple terms. The big idea is this. What if the faulty MIMO motors are the things directly responsible for messing up the centrosomes? What if this breakdown in the cell's logistics is the very thing that gives cancer cells their superpower to resist our treatments?
So here's how the chain of events might work. Step 1. The levels of the cell's tiny motors, the monos, they get all messed up. Step 2. This screws with the centrosomes, remember, the directors of operations, which causes total chaos during division. And then, step 3, this chaos is what ultimately lets the cancer cells survive the chemotherapy.
Now, that is a really powerful hypothesis, but scientists need evidence, they need proof. So by digging through years and years of existing research, a list of prime suspects started to emerge. And they found them.
The review pinpointed 16 specific MIMOs, 15 kinesins, and 1 dynein that were consistently at the wrong levels in these drug-resistant breast cancers. So boom, these are our main suspects.
But get this, it gets even more specific. The researchers found a really clear pattern. Resistance to certain chemo drugs was always linked to the same specific MIMO suspects acting up. So, for example, if the cancer was resisting tamoxifen, it was one group of faulty motors. If it was resisting docetaxel, it was a different group. This wasn't random at all. It was like they were cracking a code.
Let's just zoom in on one of these suspects, a motor called KIF14. So in cells that were resistant to the drug docetaxel, KIF14 levels were way too high. And what does it do? It basically flips a pro-survival switch for the cancer cell, a protein called AKT. And here's the proof, the smoking gun. When scientists blocked KIF14, that stay alive switch got turned off. And just like that, the cancer cells became vulnerable to the chemo again. Amazing.
So what does this all mean? Where does this leave us for the future of cancer treatment? Well, if this connection really holds up, it could open up a completely new way of thinking, a whole new path to a cure.
Okay, but it is so important to remember this part. This is a scientific hypothesis. It's a fantastic roadmap for future research, but it's built on reviewing data that was already out there. It's a powerful idea for sure, but it still has to be tested, like really rigorously, in the lab, in vitro and in vivo, before it's a proven fact. I mean, that's just how science works, one step at a time.
But there is one last really compelling piece of evidence that makes this hypothesis so strong. Remember those 16 suspect motors? Well, it turns out that 7 of them were already known to be directly involved in keeping centrosomes healthy and stable. In other words, the faulty motors weren't just suspects, they were found right at the scene of the crime.
And all of this leaves us with a pretty incredible question, doesn't it? What if we've been fighting the wrong battle all along? I mean, instead of only attacking the cancer cell directly, what if we could fix its broken internal logistics network? Could new drugs that target these tiny, faulty motors, could that be the key to finally disabling cancer's defenses for good? The investigation is far from over.